Hey everybody, so we're here for this lesson to talk about the cell cycle again, but this time our focus is on mitosis itself. So when we talk about mitosis, we have a set of specific phases that are followed. So it's broken into 5 key stages: prophase, pro-metaphase. This one, sometimes you'll hear pro-metaphase, sometimes you'll get early metaphase. People throw a few extras in here or there. Metaphase, anaphase, and telophase or telophase, and cytokinesis. Okay, so prophase, the chromatin. So notice we have a nucleus. And here's our cell, here's our nucleus. And yes, you're sketching things. So chromatin condenses. So it's going to wind up, tighten up, so it becomes chromosomes. The nucleoli, which is that one that makes ribosomes, it'll disappear from the nucleus. All the duplicated chromosomes are going to appear as it condenses. And the spindles, you'll notice, are starting to begin to form. The centromeres are moving away from each other towards opposite ends of the cells, so they'll be able to perform their role. Pro-metaphase, either late prophase or early metaphase, kind of the concept there. So the nuclear envelope that surrounded the nucleus is going to fragment and break down. And the microtubules are actually moving into that area so that they can attach to the kinetochores for all of the sister chromatids to allow for their job to take over. Then we move into metaphase, and the centrosomes are on opposite poles at this point. The chromosomes are all going to line up in the middle, and notice they're lined up so we can literally pull them in opposite directions. The microtubules are all attached, so we've lassoed all of those sister chromatids. Anaphase, they're going to move away from each other. Notice, see how they're pulling away. So the sister chromatids are separating and moving to opposite ends of the cell due to those microtubules that are now shortening. So it's like we're pulling the rope, tightening it up. This, of course, also causes and allows for the cell to elongate, because ultimately the cell is going to become 2 separate cells. That takes us into telophase, or sometimes it's pronounced telophase, and cytokinesis. Okay, so we've pulled a set of chromosomes to each end so they both match. So we have 2 daughter nuclei. So we're going to reform the nuclei with a set of chromosomes in each. The nucleoli, that's the one that makes the whole ribosomal RNA stuff, is going to reappear. The chromosomes are going to kind of relax out. They're going to be less condensed. And then cytokinesis is the physical cell division, remember? So in animals, we form what we call a cleavage furrow. Think of it as we had that thing and we tied a piece of string and we start tightening the string around it. It's a ring of actin filaments, but we're tightening the string until it splits it into 2. In plants, the vesicles that are produced by the Golgi will actually move into that middle, and they will start building a cell plate. It's like, think of, we would go in and build a wall to divide them. So we can actually track the chromosomes as we go through this process. So we started, we have mom's chromosomes, dad chromosome, and we synthesize, we make 2 copies, right? And notice, what we want you to notice is at the end, we still have identical to that original cell in terms of the chromosomes. So prophase, we have 46 chromosomes, that's 92 chromatids. Metaphase, 46 chromosomes, 92 chromatids. Anaphase, we have 92 chromosomes and 92 chromatids because we're separating out to the separate ones. Telophase, 92 and 92. And so then at the end of mitosis, when we form our separate nuclei, separate cells, they each end up with 46. So if we look at this, we can see different stages. You'll notice most of these are in interphase. Just regular blobs, relaxing, growing, developing. But we've got some different things. For example, we've got one over here that's obviously in metaphase where they're all lining up nice and neat in the middle. This one right here is obviously anaphase. You can see as they're pulling away from each other. So look through and make sure you can identify all the different phases. Okay, movement of the chromosome during anaphase would be most affected by a drug that prevents A, B, C, D, or E. So we want to stop the process in anaphase. And so the correct answer would be shortening of the microtubules. Eukaryotic chromatin is composed of DNA and proteins. Remember, we wrapped around the pencil? If a sperm cell contains 12 chromosomes, it comes from an animal that has blank chromosomes. Remember, sperm cells are gametes. Gametes have half or N. So the animal should be diploid 2N. So that should give us 24. And then one more practice-free response for you. A cell has 50 chromosomes. After mitosis and cell division, you find that one daughter cell has 49 chromosomes and the other daughter cell has 51 chromosomes. Predict one way that this could have happened. Okay, so hopefully you got a little something from that. See you in the next lesson.